Hello and welcome, dear students. Today we are going to discuss the topic Introduction to Genomic Variations. The main objectives of today's lecture are to know about the concept of genomic variations, to know about the sources of genomic variations, to know about the types of genomic variations, to know about the techniques for detecting genomic variations, and finally to understand the significance of genomic variations. Dear students, let's begin with the introduction first. Genetic variations can be defined as the genetic makeup of organisms within a population change. Genetic variation is the difference in DNA sequences between individuals within a population. So variation occurs in germ cells, that is sperm and eggs, and also in somatic cells, that is the other body cells. Variation enables organisms to adjust and adapt better according to the changing conditions of the environment. Only variation that arises in germ cells can be inherited from one individual to another and so affect population dynamics and ultimately the evolution. Mutations and recombination are major source of variation. Different kinds of variations in organisms lead to development of new species that are a better fit for the harsh environmental condition. Variation can be seen at every genetic level, that is in the DNA, in the genes, in the chromosomes, in the proteins, and in the function of proteins as well. Genomic variation can be defined as the DNA sequence differences among individuals or populations. Some variants influence biological function, that is such as a mutation that causes a genetic disease, while others have no biological functional effects at all. By understanding genomic variation in families, in populations, we can know how our genetic makeup varies between individuals. It may help us to understand why even within families people have different blood groups and eye colors. Genomic variation may also be the reason why some populations or persons within a certain population may have higher risk of certain diseases compared to others in the same population. Variation in the genetic material of an organism is depicted in its phenotype. Almost every trait of an individual is affected by genetic variation. So variations in the eye color and hair color, ear lobes, height, etc. are due to the genetic differences between individuals. Genetic variation is an important evolutionary force as it provides the diversity within and between the populations. Genetic variability makes an organism more adapted to its environment. Variations result in different forms of gene, that's called alleles or alternative forms of a gene. For example, if a gene with capital A is mutated to a gene with a small a, and if the new allele, that is the small a, has some selective advantage for the individual either through fitness or fecundity, then the mutated allele will be transmitted to the next generation over a period of time. On the other hand, if the new allele is deleterious, then it would be selectively eliminated as the offspring carrying it will not survive. Pre-existing advantageous mutations, when selected, will result in observation of new phenotypes. This process is called natural selection. Natural selection brings about change in the gene frequency. Over a few generations, this would result in speciation, that is the creation of new species of organisms. Hence, disturbance in genetic equilibrium or Hardy-Weinberg equilibrium, that is the change of frequency of alleles in a population, would lead to evolution. Dear students, let's now discuss the source of genomic variations. Mutations, gene flow, and sexual reproduction are three primary sources of genetic variation. Let's discuss first the mutations. Mutation is a process which produces altered DNA sequences. It may be induced by radiations and chemical mutagens in the environment or insult from endogenous reactive oxygen species, that's ROS, or occur spontaneously due to endogenous factors like errors in chromosome segregation, recombination, DNA replication, and repair mechanisms, and also spontaneous chemical damage to the DNA. Mutations are unavoidable. They may have adverse effects on organism's health and survival. Sometimes they may have no obvious effects, or rarely they may be beneficial for the bearer. Dear students, let's now discuss another source of variation, that is the gene flow. So gene flow is the transfer of genes from one population to another and thus affects the allele frequency. The rate of gene flow is directly proportional to the mobility of an individual and is limited by geographical barriers which may be natural or man-made. Cross-species hybridization in plants to generate new and improved varieties and gene transfer from bacteria or virus to new hosts lead to genomic variations in individuals. Dear students, let's now discuss another source of genomic variation, that is the sexual reproduction. So somatic cells divide and multiply by mitosis that produce daughter cells identical to that of the parent cell, whereas during gamete formation a special type of cell division, that is meiosis, also known as reductional division, results in newer combinations of parental genotypes by the phenomena of crossing over and independent assortment of homologous chromosomes, thus generating unique genotypes followed by random fertilization. So crossing over acts as a raw material for the evolution to occur. Sexually reproducing organisms are more diverse than the asexually reproducing ones. Dear students, let's now discuss the types of genomic or genetic variants. Genetic variations are basically of three types. One is single nucleotide variations or SNVs. Then insertion, deletion, that is indels, and structural variations. These variations may be rare or common depending upon their frequency in the population and pathogenic or non-pathogenic. Let us first discuss the single nucleotide variations or SNVs. SNVs or single nucleotide variations are the most common type of all variations where a nucleotide is replaced by another one. It may be either transition, that is purine to purine nitrogenous base, or pyrimidine to pyrimidine nitrogenous base, or transversion where a purine is replaced by a pyrimidine, or vice versa. When a single nucleotide variation is common in a population, or we can say its frequency is less than 1%, then it is known as single nucleotide polymorphism, or SNP. SNPs may fall within the coding or non-coding regions of genes, that is DNA, or in the intergenic regions between the genes. So coding region SNPs may be either synonymous, that is no change in amino acid sequence, or non-synonymous, that is change in amino acid sequence. SNPs are usually selectively neutral, but they often make an individual predisposed to certain diseases. For example, SNPs in MTHFR, that is methylene tetrahydrofolate reductase enzyme, predispose an individual to cardiovascular diseases, cancer, and neural tube defects. SNPs also confer a selective advantage to individuals harboring them. For example, Duffy blood group allele, FY small a small b, that is Duffy null phenotype, protects against malaria. So SNPs are cataloged in a public database represented by this particular address. Coding regions, that's SNVs, may affect the structural and functional aspects of a protein. They are usually deleterious and present less frequently in population, that's about 1%. They are known as rare variants. So rare variants which are clinically significant are cataloged in clinical variation database represented by this address. SNPs present in non-coding regions are also reported to influence gene splicing and transcription factor binding. The functional effects of variants on the protein structure and function can be predicted using various in silico tools or bioinformatic tools like SIFT or Polyphen2 or mutation taster, etc. Dear students, let's now discuss insertions and deletions. Both are collectively called as indels. So indels refer to DNA mutations involving insertion and deletion of one or more base pairs. These can have devastating effects on the gene because either translation of the gene is frame shifted or amino acids are deleted. For example, 3BP deletion, that is DF508, in coding region of the CFTR, that is cystic fibrosis transmembrane conductance regulator gene, is the most common cause of cystic fibrosis. However, non-pathogenic indel are often useful in mapping common ancestry in kinship and human origin studies because chances of two mutations of the same length occurring in the same genomic position are negligible. Dear students, let's now discuss duplication. A duplication occurs when a stretch of one or more nucleotides in a gene is copied and repeated next to the original DNA sequence. This type of variant may alter the function of the protein made from the gene. The type 1A form of Charcot-Marie-Tooth disease is an example of an inherited human genetic disease that is caused by a gene duplication. Dear students, let's now discuss the inversion. An inversion changes more than one nucleotides in a gene by replacing the original sequence with the same sequence in reverse order. For example, hemophilia A genetic bleeding disorder, is often caused by chromosomal inversions that involve a portion of the blood coagulation factor 8 gene that encodes one of key enzymes in blood clotting. Dear students, let's now discuss the frame shift mutations. A reading frame consists of a group of 3 nucleotides that each code for one amino acids. That's the codon. A frame shift variant occurs when there is an addition or loss of a nucleotides that shifts the grouping and changes the code for all downstream amino acids. The resulting protein is usually non-functional. Insertions, deletions, and duplications can all be frame shift variants. Dear students, let's now discuss repeat expansion. Some regions of DNA contain short sequences of nucleotides that are repeated a number of times in a row. For example, a trinucleotide repeat is made up of sequences of 3 nucleotides, and a tetranucleotide repeat is made up of sequences of 4 nucleotides. So a repeat expansion is a variant that increases the number of times that the short DNA sequence is repeated. This type of variant can cause the resulting protein to function improperly. Dear students, let's now discuss the structural variations. Structural variation refers to a large-scale structural differences in the DNA originated mainly due to chromosomal rearrangements of deletion, duplication, novel sequence insertion, or inversion. They include copy number variations, variable number of tandem repeats, and chromosomal rearrangements. Dear students, let's first discuss the copy number variations or CNVs. A CNV or copy number variation can be simple in structure such as tandem duplication or may involve complex gain and loss of homologous sequences at multiple sites in the genome. CNVs are genomic structural variants in which the number of copies of a particular segment is different in two or more different genomes as compared to a reference genome. Dear students, let's now discuss variable number of tandem repeats or VNTRs. These variations involve change in the number of repeated DNA sequences arranged in a tandem array. So these are highly heterogeneous group of loci. These are multi-allelic markers while SNPs are generally biallelic. They are classified according to the size and number of their repeat units. They are sometimes considered as neutral markers. But there are many instances among genetic variation where various classes of VNTRs which show phenotypic effects. So VNTRs can be microsatellite, mini-satellite, satellite or telomere repeat arrays. Dear students, let's now discuss the microsatellites. They are also known as short tandem repeats or STRs or simple sequence repeats or SSRs, having 1 to 6 base pair long repeat units. Their copy number varies from 10 to 30. Microsatellites with some specific repeat units show clustering, but most are distributed throughout the genome, which is useful in linkage analysis. The most common microsatellite is CAN repeats, that is mutation at these regions may involve either increase or decrease in repeat units. Microsatellites are reported to be associated with many human diseases like Fragile X syndrome or Huntington's disease, etc. Dear students, let's now discuss the mini-satellites. Mini-satellites are tandem arrays of repeat units around 10 to 60 base pairs long with a copy number of 5 to 50. They are generally GC, that is guanosine and cytosine rich repeats. They are among most dynamic loci in our genome. Like most of the microsatellites, these are considered to be neutral markers, but there are examples that they play essential roles. Dear students, let's now discuss the satellite. They are also called macrosatellites. They are large tandem repeat arrays spanning hundreds of kilobases to megabases composed of repeat units of wide range of sizes that can display a higher order structure. Alpha satellite or alphoid DNA is a good example of the satellite. It is 170 base pairs in length. Their detailed structure is difficult to study due to their size and repetitive nature. Dear students, let's now discuss telomere repeat arrays. Telomeres are the structures present at the end of the chromosomes having tandem arrays of hexanucleotide repeat TTAGGG, typically 10 to 15 kilobases in length. One end of this array is contiguous with subtelomeric DNA, while other is the true end of the chromosome. Dear students, let's now discuss the chromosomal rearrangements. Chromosomal rearrangement involves deletions, duplications, inversions, and translocations, which are caused by either breakage of DNA double helixes at two different points, followed by rejoining of the broken ends or through crossing over between repetitive DNA sequences to produce a new chromosomal arrangement of genes. Chromosomal rearrangements may lead to disruption of genes and are implicated in many human disorders, particularly cancer, like chronic myelogenous leukemia. where balanced translocation results in fusion of the breakpoint cluster region with the proto-oncogene tyrosine kinase, leading to the formation of BCR-ABL fusion transcript, which constitutively activates tyrosine kinase that can transform cells and inhibit apoptosis, that is the programmed cell death. Chromosomal rearrangement is also a common mode of speciation, that is the formation of new species, mainly through post-mating reproductive isolation. For example, inversion resulted in species diversity between two closely related Drosophila species, Drosophila pseudo-obscura and Drosophila persimilis. Gene duplication also causes bar eye phenotype in Drosophila melanogaster, that's the fruit fly. Dear students, this was all about genetic variations and their types. Now let's discuss the techniques for detecting genomic variations or genetic variations. Genotyping provides a measurement of the genetic variation between members of a species. Genomic variations are often found to be the etiology of many human diseases and are of particular interest in pharmacogenomics. The increase in interest in the genomic variation has been reflected by the rapid development of a diverse range of single nucleotide polymorphism genotyping methods. Dear students, let's discuss the applications of genomic variations. First, screening for genetic diseases. Genomic variations can cause many diseases in humans. These diseases are usually rare but sometimes may be fatal. And an example is cystic fibrosis, caused by mutations in the CFTR gene. Many common diseases such as heart diseases and cancer are genetically complex, with alleles from several genes contributing to the disease. Linkage and association studies are used to conduct genetic tests for screening of diseases, which involve profiling of disease-causing variants. Dear students, let's now discuss pharmacogenomics. Genetic variations or genomic variation in patients make them respond differently to a medication. In the future, the most appropriate drug for an individual could be determined in advance of treatment by analyzing a patient's SNP profile, and such kind of treatments which are specific for an individual are called personalized medicines or precision medicines. Dear students, let's now discuss biological markers. Often, genomic variations tend to be relatively stable genetically. They serve as excellent biological markers. Constructing a chromosome map that shows the position of known genes with genetic markers allows researchers to study and pinpoint disease traits. Use of genetic variations in forensic technologies. Genetic profiling of biological material has become one of the most powerful tools for solving parental disputes and in criminal investigations. Traditional methods in DNA fingerprinting are now replaced by polymerase chain reaction or PCR-based technology, which detects very short polymorphic stretches of DNA. Dear students, now let us study what is the significance of human genetic variation. Almost all human genetic variation is relatively insignificant biologically, that is, it has no adaptive significance. But some variation, for example, a neutral mutation alters the amino acid sequence of resulting protein but produces no detectable change in its function. Other variation, for example, a silent mutation does not even change the amino acid sequence. Furthermore, only a small percentage of the DNA sequence in the human genome are coding sequences that are ultimately translated into proteins, and it's less than 1.5%, or regulatory sequences that can influence the level timing, and tissue specificity of gene expression. Differences that occur elsewhere in the DNA in the vast majority of the DNA that has no known function have no impact. Some genetic variation, however, can be positive, providing an advantage in changing environments. The classical example from the high school biology curriculum is the mutation for sickle cell anemia, where the hemoglobin in the RBCs changed its structure, which is heterozygous state provides a selective advantage in areas where malaria is endemic. More recent examples include mutations in the CCR5 gene that appears to provide protection against AIDS. The CCR5 gene encodes a protein on the surface of human immune cells. HIV, the virus that causes AIDS, infects immune cells by binding to this protein and another protein on the surface of those cells. Mutations in the CCR5 gene that alters its level of expression or the structure of the resulting protein can decrease HIV infection. Early research on one genetic variant indicates that it may have risen to high frequency in Northern Europe about 700 years ago, at about the time of the European epidemic of bubonic plague. This finding has led to some scientists to hypothesize that the CCR5 mutation may have provided protection against infection by Yersinia pestis, the bacterium that causes plague. The fact that HIV and Yersinia pestis, which causes plague, both infect macrophages supports the argument for selective advantage of this genetic variant. The sickle cell and AIDS plague stories remind us that the biological significance of genetic variation depends on the environment in which genes are expressed. It also reminds us that differential selection and evolution would not proceed in the absence of genetic variation within a species. Some genetic variations, of course, is associated with disease, as classical single gene disorders such as sickle cell disease, cystic fibrosis, and Duchenne's muscular dystrophy remind us. Increasingly, research also is uncovering genetic variations associated with the more common diseases that are among the major causes of sickle cell anemia and death in developed countries. Diseases such as heart diseases, cancer, diabetes, and psychiatric disorders such as schizophrenia and bipolar diseases. Whereas disorders such as cystic fibrosis or Huntington's disease result from the effects of a mutation in a single gene and are evident in virtually all environments. The more common disease results from the interaction of multiple genes and environmental variables. Such diseases therefore are termed polygenic and multifactorial. In fact, the vast majority of human traits, diseases or otherwise, are multifactorial indeed. The genetic distinctions between relatively rare single gene disorders and the more common multifactorial diseases are significant. Genetic variations that underlie single gene disorders generally are relatively recent, and they often have a major determinantal impact, disrupting homeostasis in significant ways. Such disorders also generally exact their toll early in life, often before the end of childhood. In contrast, the genetic variations that underlie common multifactorial diseases generally are of older origin and have a smaller, more gradual effect on homeostasis. They also generally have their onset in adulthood. The last two characteristics make the ability to detect genetic variations that predispose or increase risk of common disease especially valuable, because people have time to modify their behavior in ways that can reduce the likelihood that the disease will develop, even against a background of genetic predisposition. Dear students, that was all about today's lecture, Introduction to Genomic Variations. Hope you have understood it well. Thank you and goodbye.